Hey guys, here is our lecture for 2.3a, carbohydrates. Now we're going to split this up into two lectures. The first one is going to be about carbohydrates, and then 2.3b is actually about lipids. So let's get started. So let's go back to like freshman year for a minute and do a review of basic info about carbohydrates. So carbohydrates, they're one of the four categories of organic molecules, which are carbs, lipids, proteins, and nucleic acids. Some carbohydrates are used for energy, and some are used for structure. When you have a carbohydrate, you always have this 1-to-1 ratio of carbon to hydrogen to oxygen. You may remember the whole cho, chan, chomp thing. Well, this is our first cho: carbon, hydrogen, oxygen, CHO. Glucose is a good example of a carbohydrate monomer, and you can tell it's glucose because it's got 6 sides, right? We have that carbon ring, and this ring is formed because carbon wants to make 4 bonds. So if you look around this carbon ring, every carbon molecule is bonded with 4 other molecules, 4 other atoms. We also have fructose, which has the same chemical formula, C6H12O6, but its shape is different. It's only 5-sided. So be aware, just because two molecules have the same chemical formula doesn't mean they're the same thing. Your shape is very important in biology. Your shape is your function. Your shape is your job. So, if a carbohydrate has 4 carbons, how many hydrogen and oxygen would it contain? Let's see. If you said 8 hydrogen and 4 oxygen, you are correct. Okay. Now let's get into our understandings. Understanding number 1. And remember, these understandings, these are from the applications, skills, and understandings list. That's your checklist of what you have to know for this section for 2.3. Understanding number 1. Monosaccharide monomers are linked together by condensation reactions to form disaccharides, and polysaccharide polymers are broken down via hydrolysis. All right, so let's translate that into English. Monosaccharides are monomers. Mono means one. So mono means one ring. 2 monosaccharides together form a disaccharide. Di means 2. So here we have a monosaccharide, right? One ring. And here we have a disaccharide. 2 rings. One here, 2 here. Disaccharide. Now, you do have to know certain molecules. For this course, the 3 monosaccharides that you need to know, well, there's more than 3, but there's ribose too. In addition to ribose, you need to know glucose, galactose, and fructose. For disaccharides, you need to know maltose, lactose, and sucrose. For polysaccharides, you need to know starch, glycogen, and cellulose. These 9 molecules will keep coming up in our discussions throughout the next 2 years. In fact, the seniors right now are learning about how starch, which is also called, we use amylose, and they are doing labs right now that look at how you actually execute the process of going from this polysaccharide, right, using hydrolysis and enzymes to end up with monosaccharides here. And they're doing that. That's what all the lab equipment was last week. So these 9 will keep coming up. So let's talk about them in detail. So first, let's talk about sucrose. Sucrose, again, disaccharide, because it's got 2 rings. 1, 2. We form sucrose by putting together one glucose molecule and one fructose molecule. That condensation reaction pulls a hydroxyl group from the glucose and a hydrogen from the fructose, and water comes out. That's why it's a dehydration synthesis reaction. The result is a disaccharide, sucrose, right, with a glycosidic linkage. That's just the name for the bond, glycosidic linkage. Sucrose is table sugar. Okay, so now let's go to maltose. Maltose is also a disaccharide. Should we write disaccharide here just so that we're clear? Maybe we should. Sucrose is a disaccharide, right? So we have monosaccharide, because it's one ring, plus monosaccharide, because it's one ring, equals disaccharide, because it's two rings linked together. Oops. Okay, so now let's look at maltose. Maltose is another disaccharide that's formed from two molecules of glucose. The result is maltose. So now we have two six-ringed glucose molecules that are held together by an oxygen and a glycosidic linkage. So if you're ever asked what kind of bond holds glucose monomers together or carbohydrate monomers together, it's a glycosidic linkage. Okay, next is lactose. Now, this is the sugar in milk that some people can't process if you are lactose intolerant. This is actually it. This is the big bad guy right here. So we have a galactose and a glucose molecule. Dehydration synthesis combines these molecules, releases a molecule of water to form lactose. And we'll talk all about lactase and how we break that down a little bit later. So you'll notice this has a D in front of it, D-galactose and D-glucose. That is the orientation of the molecule. So molecules are either left-handed or right-handed, depending on which way they face. We don't really need to know a whole much about that. All you need to know really is that lactose is galactose and glucose put together. So if I were going to write this like I did before, I would probably write, let's see, I'll do it over here. Galactose plus glucose equals lactose. Right? You have your monomers. One ring, links together to form two rings. Two monosaccharides linked together to form a disaccharide. So in review, let's take a look at those reactions and how they happen. When you're putting molecules together, dehydration synthesis, right? Tell me, is this catabolic or anabolic? You should be able to tell me. You know what? I'll ask a question in a second. So let's look at dehydration synthesis, right? So we have a six-sided, this looks like a glucose molecule, right? And then we have a five-sided ribose. Let's see what happens. That water molecule drops out, and here we have our disaccharide. Now, hydrolysis is the opposite. We're going to start with our disaccharide, and we're going to add water and break this glycosidic linkage right here. So that same water molecule that we dropped out, we're going to put it back in, and we're going to use it hydrolysis. Hydro, water, lysis means to break, right? We're literally breaking with water. There's our water molecule. And it's going to break that disaccharide up back into monomers again, monosaccharides. Okay, those were the monosaccharides and disaccharides. Now let's talk about our polysaccharides. Poly means many, right? So now we're not talking about just one or two little rings. We're talking about tens, hundreds, thousands, hundreds of thousands. So glycogen is a starch that's found only in animals. And you can see it's highly branched. Remember, energy is stored in chemical bonds. Energy is stored in chemical bonds. Right? So, more bonds equals more energy. So, it's not surprising that energy storage molecules like glycogen have lots and lots and lots of monomers. Their whole point is to store energy in those bonds between those monosaccharides. Now, like I said, glycogen is in animals. Starch you tend to find in plants. And then there's cellulose. Now, cellulose is not energy storage. It's more for structure. So this is what gives plants its structure. This is also why humans can't eat grass, right? So different animals like cows and horses and deer, they have special enzymes that break down cellulose. Humans do not have that capability. We do not possess the gene that codes for the enzyme that breaks down cellulose. So like when you take fiber pills, what you're basically doing is taking cellulose and it helps kind of like push everything through your GI tract because your body doesn't have the ability to break it down. In more complex language, cellulose is a polysaccharide made up of straight chains of beta glucose units. And the hydrogen bonds are going to form between their OH groups. Okay, so you're probably like, WTF is this nonsense? I don't even know what I'm seeing here. Think back to when we talked about water. We talked about water as being a Mickey Mouse head, right? And you have, that's a bad Mickey Mouse head. You had a oxygen and you had hydrogen ears. And the ears are positive, and the head was negative. And between two different water molecules, you had hydrogen bonding, right? So this hydrogen was going to be attracted to that oxygen. And that's what gives water a bunch of its special properties, right? That's why it takes more energy to evaporate. That's why water is less dense when it freezes. All those things we talked about last time. Well, cellulose also forms, cellulose also forms hydrogen bonds. So I know this looks really funky, but this thing here is one glucose molecule, and this thing here is one glucose molecule. And see all these little dashed lines between not the glycosidic linkages, right? The glycosidic linkages that actually hold the glucose molecules together is right here. So that's like your regular covalent bond where you store lots of energy. But look at all these little dashed lines everywhere. These little dashed lines, these are hydrogen bonds. Look, they're all between an oxygen and a hydrogen. It's all these hydrogens checking out all these other oxygen side chicks. These extra hydrogen bonds help make cellulose very rigid and a good structural molecule. So while there is a ton of energy that's stored in cellulose in each of those covalent bonds, or the the glycosidic linkages here, right? There's lots of energy stored in each one of these. The purpose of this molecule isn't to store energy. The purpose of this molecule is structure. And these hydrogen bonds, all these little dashed lines, are extra braces that help hold this molecule together. So these cellulose chains group together to form microfibril bundles, and they have a high tensile strength, meaning it's very hard to break them. They also make up the basis of cell walls. So when we're talking about, like, how plants have cell walls, cellulose is the actual stuff that makes up cell walls. And that makes sense. This gives plant cells, the ability to withstand pressure, internal pressure. Think about if you're a cell in a tree and you're towards the bottom, think about how much weight is on top of you, especially if you're like a redwood tree, right? That's huge. You can think of these cellulose boxes around these cell walls around plant cells as like tiny little skeletons that help that cell maintain pressure. Now, when we go to starch, right, starch polysaccharides, they form when alpha glucose molecules link together. Now, these guys are a little bit different. They're oriented in the same way, which causes them to curve into a helical structure, kind of like DNA. In fact, remember DNA has a sugar backbone? That's going to look familiar later on. Two types of plant starches that you should know are amylose and amylopectin. Their job is to store energy. So, the more bonds, right, the more glycosidic linkages you have, the more energy is stored. Amylopectin is branched and it helps stores lots of energy, whereas amylose is just one long chain. So it stores less energy. So this is amylose. This is amylopectin. Amylose is like a branch, single string branch. Amylopectin has more branches. Here's glycogen. This is our animal starch. Animal. And it's similar to that branch form of starches in plants. You can see here are all these glycosidic linkages, right? But glycogen is huge, much bigger than amylopectin or amylose. And when I say huge, I mean like super huge. Like that doesn't even show how really big it is. All these extra branches gives you more access to glucose because enzymes can reach them in a smaller area. So think of it this way. If I'm an enzyme, there's lots of places for me to attack and break bonds, as opposed to if it was just one long chain. So glycogen is a really functional and useful molecule for storing lots of energy in a small space. Kind of like when you wad all your clothes up together in a ball. Okay, so we have some links here. And these are the same links that you looked at before to help you compare some of the shapes of your starches. So when you did that visualization exercise, that's what you were looking at. You were looking at cellulose. You were looking at amylose. You were looking at amylopectin. You were looking at glycogen. I encourage you to use these links and go back and look again now that we've talked about the functional differences and the uses of these starches, right? And these carbohydrates. Some are for energy and some are for structure. Some are for structure, not infrastructure. There we go. Here is a nice handy dandy little table that reviews all the different molecules that we talked about today. We talked about your monomers, glucose, fructose, and galactose. You can see they're mono. This means monomers, or I'm sorry, same thing, monomers or polymers, right? Monosaccharides, disaccharides, polysaccharides. So these guys are monosaccharides. And you see this OH up, down, OH up, up. That refers to whether the hydroxyl groups are pointing up or down. You can go back and look at that. Here is the chemical formula. Here's the sides, right? 6-sided, 5-sided, 6-sided, and the purpose. Glucose is usually energy, right? Fructose is energy, galactose is energy. Now you have maltose, which is glucose plus glucose. It's a disaccharide. Here's its chemical formula. It's two monomers, so we don't have the number of sides there. And its purpose is to store energy. Sucrose, table sugar, is glucose plus fructose. It's a disaccharide. Here's its formula, and it's used to store energy. Glucose plus galactose equals lactose. This is a sugar found primarily in mammal milk. It's a disaccharide. Here's the chemical formula, and it's used for energy. Now we have our complex starches, right? We have cellulose, which is a polysaccharide, and it's used for structural purposes in plants, particularly the cell wall. There's amylose, which is a polysaccharide. It's linear, right? It's a long chain of glucose monomers, and its purpose is energy storage in plants. Then there's amylopectin, which is a polysaccharide. It's branched. It's got all those branches. It stores more energy than amylose, and it's used in plants. And finally, glycogen, which is the polysaccharide that you find in animals. It's very highly branched, right? And its purpose is energy storage in animals. So there it is. There are your notes and your lecture for carbohydrates. We're going to do some fun stuff with this in a couple of, I think a week or two. We're going to do some labs and we're going to watch how you go from one to the other using certain chemicals and dialysis tubing. It's going to be a whole lot of fun. So use this to make sure you have good notes. And then I'll see you in a few minutes for lecture part B, lipids. Thanks for watching.